So it's a part of the hindbrain. So when we talk about hindbrain, that means it's the pons, the medulla, and the cerebellum form the hindbrain, or we call it the rhombencephalon. And in that, we have the cavity of the hindbrain is the fourth ventricle. So the cerebellum forms the roof of the fourth ventricle, while the pons and the medulla form the floor of the fourth ventricle. I'll go on to that later. The cerebellum, where does it lie? We've already done the craniocerebellar cerebral relations in the first practical, where the cerebellum lies in the posterior cranial fossa. That's in the occipital bone. And if you see this posterior cranial fossa, if you remember very clearly, it has an internal occipital protuberance, a transverse sulcus, and then there are two transverse sulcus, in which is attached the tentorium cerebelli. So the tentorium cerebelli, it's a horizontal fold of dura mater like a tent, which separates the cerebral cortex from the cerebellar. So the cerebrum and cerebellum are separated by this double layered meninges, which is called the tentorium cerebelli. So that is the tentorium cerebelli, which is separating between the cerebrum and the cerebellum. So what is the function of the cerebellum? The function of the cerebellum is it controls posture and voluntary movements of the same side of the body. It works like some of these sensory tracts which you have studied, they are usually occupying the opposite side of the body. But this is a part which is involved in influencing the same side of the body. That is, it maintains the posture, the movements, and the equilibrium of the body. Unconsciously influences the smooth contraction of voluntary muscles and coordinates unconsciously influences the smooth muscle contraction of the voluntary muscles and coordinates their actions together with relaxations of the antagonist. So do you understand that? So if I'm flexing my arm to get something, what's happening? The extensors are relaxing. So this kind of coordination between agonist and antagonist, when they are taking place, we are doing it reflexly, we are doing it unconsciously. We do not know what we are doing. When we have to pick up a glass to drink water, our muscles are all acting together. There's no jerk, there's no discrepancy in the movements. It's a smooth flowing movement which is helping us do these activities, these motor activities. So it is a regulation, so it is helping the regulation between nerve and muscle impulses. Cerebellum has no direct pathway to the lower motor neuron, and it is getting influenced by the cerebral cortex and brainstem. So it's only inputs which are coming, it's a processor in between. So the inputs which are coming from the higher centers and the brainstem are coming to the cerebellum, getting interpreted, and then goes down to the spinal cord by the pathways. So this is just to show you a section of the cut section of the brain. This is the medial surface of the cerebral hemisphere, and we see the part of the brainstem, and we also see part of the cerebellum. Now what do you see out here? I'm just now acquainting you with this, since you have already done the brainstem, acquainting you with these pictures, because when we do the next class, these pictures are going to come across and we have to identify the structures. Look at the cerebral cortex. If you see, as my cursor goes, what do you see? They are depressions and elevation. These are the sulci and gyri. Just at this part, there is a bundle of white matter which is called the corpus callosum. And if we come here, there is a small bundle again. This corpus callosum is a part which is called a commissural fibers, which are connecting the two sides of cerebral hemispheres. They are connecting the two sides. This is another connection between the two sides, which is called the anterior commissure. Then we have this is the lamina terminalis. This part out here, if you see, is the optic chiasma, and we have the mammillary infundibulum stalk. So this is the part which we will be doing when we do diencephalon, but just see this corpus callosum and this bundle of fiber that you see out here is the fornix. This portion that you see is the part of the diencephalon, which is the hypothalamus and thalamus, and cavity of the cerebral cortex is the lateral ventricle. This would be the lateral ventricle. Inside this, there is a fluid-filled cavity. The cavity for the diencephalon is the third ventricle. It's a slit, not a very big cavity. The cavity of the midbrain is again a thin cavity which is called the aqueduct of Sylvius or the cerebral aqueduct. So this portion that you see out here is the midbrain. You can see the superior and inferior colliculus, and out here this is the crus cerebri. So we have tectum, tegmentum and crus cerebri in the midbrain. This is the cavity, the cerebral aqueduct, and then when you come down, this is the pons and this is the medulla. And the pons of the medulla are forming the floor of the fourth ventricle, which is looking like a rhomboid shape, or we say a triangular. Here it looks like a triangle on this shape, and the roof of this is formed by the cerebellum. Now look at the cerebellum. The cerebellum is also folded on itself like the cerebral cortex, but the folding out here is very different. They form horizontal fissures. Look at the surface. They form fissures out here. Instead of the sulci gyri, they are parallel fissures which are dividing the cortex, so it increases the surface area. And the white matter which is present forms like a branching tree, which is called the Arbor Vitae appearance. It looks as if branches of the tree, so that is an Arbor Vitae appearance. Now the cerebellum, if you look carefully, is connected to the brain stem by peduncles. These are nothing but bundles of white matter which are connecting them. So they are forming the channels through which afferents and efferents will travel. So to the midbrain, it is connected by a peduncle which is called the superior cerebellar peduncle. This part where I'm showing my cursor is the superior cerebellar peduncle. The part with the pons is the middle cerebellar peduncle, and the part with the medulla is called the inferior cerebellar peduncle. These are the cut sections of the peduncles. The superior cut section is with red, middle with green. That's with pons, and with the medulla, it's the inferior cerebellar peduncle. And the part of it has been reflected away to show you the floor of the fourth ventricle, which is formed by the upper part by the pons and lower part by the medulla. So this is a very clear picture showing you the posterior part of the midbrain. This is the superior and the inferior colliculus, which are called the corpora quadrigemina, or we also call them the tectal plate. This is the cerebral aqueduct. The CSF, which is present in the midbrain, lies in this. This is the mesencephalon. Just above the corpora or the tectal plate or the colliculi lies this small gland which is called the pineal gland. The pineal gland, And the pineal gland is also connected by two commissures. One is called the anterior, one called the posterior commissure, and the posterior commissure, and this one is called the habenular commissure. We'll be doing them later, but just remember that this is where it is. So if there's a pointer sometime, you will be able to see. And anteriorly out here, you see that there is a swollen part. This is called the mammillary body, a part of the hypothalamus. I'll be doing it when I do diencephalon with you. So this is the pons. Look at the fibers of the pons, transverse fibers. So the pons sends fibers to the cerebellum by middle cerebellar peduncle. Midbrain is connected by the superior cerebellar peduncle and medulla connected by the inferior cerebellar peduncle. So these white matter which are going in are actually dividing and forming this branching pattern. And outside you see the gray matter. Look at the shape of this, and what happens out here, as this is forming, there will be small apertures or small foramens between the fourth ventricle and that's in the roof, which would be called the median aperture, and there are two lateral apertures out here. So these are the ones which would be allowing CSF to come out into the subarachnoid space. So from here they'll be going out, and then they come onto the subarachnoid space, onto the spinal cord, and then they'll be going around the brain and cerebellum. So CSF circulation takes place. Now those were all diagrammatic pictures. Now let's look at the cerebellum has been removed from the brainstem, and it has been shown like this. So when we look at the cerebellum, look at the structure, what do you see? It has horizontal fissures. It has horizontal fissures. Look at its structure. It is a structure which has two surfaces, a superior surface and an inferior surface. If you look at the superior surface, what do you see in the center? You find that there's something like an earthworm. That's why it looks like a vermion. That's why it's called the median part is called the vermis, like a worm. So it is called the vermion. And laterally, these expanded parts are called the cerebellar hemispheres. The superior surface is in fact convex. The vermis almost merges with the cerebellum hemispheres. While the inferior surface, if you see, it has a depression out here, and this depression is called the, so this vermis, if you see it from the inferior surface, has a depression, and this is called the vallicula. This is called the vallicula. From that, one would be able to say which is the superior surface and which is the inferior surface. Right? There are number of fissures in this. There are number of fissures which are present in the cerebellum, but for our study, we will only concentrate. I'm not going to tell you about all the fissures. I'll be talking about only 3 fissures. So you please concentrate on that. When I'm talking about only 3 fissures, one out here is the primary fissure, or it's called the fissura prima. If you look at this fissure, as my cursor goes, you'll find it forming a U-shaped fissure with some amount of vermis and some amount of cerebellum with it, hemisphere of the cerebellum with it. The part which is anterior to this primary fissure is called the anterior lobe. The part which is posterior to the fissure is called posterior lobe. So the primary fissure is dividing the cerebellum into an anterior part and a posterior part. That means the anterior part is called the anterior lobe, the posterior part is called the posterior lobe. In some books you'll find that it is also mentioned as the middle lobe. But I would try to say posterior lobe for convenience, because I find it more convenient to say that this is the posterior lobe. And there is another fissure which is separating a part of the inferior surface of the cerebellum from the rest of the cerebellum, which is called the dorsolateral, or we say the posterolateral fissure. Look at this. This is a very thin layer of cerebellum, which is called the, and there is some, if you see, a small grape-like or mulberry-like structure, that is the flocculo-nodular lobe. The flocculo-nodular lobe, the nodule in the center, and this is called the flocculus. So it is separated from the entire cerebellum by a fissure which is called the dorsolateral fissure. This that you see out here is the vallicula, because this is the depression and this is the inferior vermis. Look at this. This is the inferior vermis. There is another fissure between the superior surface and the inferior surfaces, which cuts it here at this part. It's not labeled in this diagram. In the later diagrams it's labeled, which is called the horizontal fissure. But the horizontal fissure is not dividing it into any lobe. It's only a demarcation between the superior surface and the inferior surface, right? So now let's look at this. Here I have mentioned this. This is the horizontal fissure between the superior. So two surfaces, the morphology of the cerebellum is it had two surfaces, a superior surface and an inferior surface. This is the superior surface, almost convex, right? The part in front of the primary fissure is the anterior lobe. The part behind the primary fissure is the posterior lobe. So this is the fissura prima, and then there'll be another small fissure between the flocculus. This is the nodule, and that is the flocculus. There will be the posterolateral fissure. So posterior lobe is very big, while anterior lobe is smaller. So the anterior lobe which is present, it only occupies 2/3 of the superior surface, while posterior lobe is occupying the entire posterior inferior surface and this part of the superior surface. Now look at this picture. If you look at this picture, this is from Snell's Neuroanatomy. You'll find in all textbooks that there is a horizontal fissure. That means what has happened out here? The cerebellum has been opened up like the pages of a book. Horizontal fissure के आगे जो है, that is the anterior superior surface, और पीछे जो है, that is the posterior surface. So this is the posterior surface and this is the anterior surface. And if you look at this carefully now, this is the fissura prima, and in front of whatever is there has been marked pink. That means the anterior lobe has been marked pink. The posterior lobe is brown, and the flocculo-nodular lobe, which is separated from the rest of the cerebellum by the uvulonodular fissure, or it's also called the posterolateral fissure, is been marked. What color? It's been blue. So this is what it is. And if we look at this vermis, each part of the vermis, now this is the lingula. The first part of the vermis, this top end is called the lingula, right? The lingula has no lateral hemispheres. After that, we have the vermis has been divided in such a way that if I say this part of the vermis is the central lobule, the lateral part of the cerebellar hemisphere is called ala. This portion is called culmen, and the hemisphere with it is called the quadrangular lobe. This portion of the vermis is called declive, the median part, and the cerebellar hemisphere is called lobulus simplex. This is folium. This is called the superior semilunar lobule. This is a part of the superior surface. Now this is the inferior surface of the cerebellum. I hope you are able to understand when I am saying that we have opened up the cerebellum. So this is the tuber, and this is the inferior semilunar tubule. This is the pyramid, and this is called the part of the cerebellum is called the biventral lobule. This is the uvula, and the lateral portion is called tonsils. Now, what is here? There are a number of terms which have come up, but I would request you do not get perturbed with so many terms coming up. Whatever terms I'm going to tell you, just remember those. एक आपको remember करना है the floccular nodular lobe, which is very easy to remember. This part is the floccular nodular lobe, the lingula, right? The cerebellar tonsils, remember that. The rest, just remember, in front of the fissura prima is the anterior lobe, the rest is all the posterior lobe. Just remember that for the time being. Is it clear? After that, we will go on to the next. So if there are any questions. So, right, no questions. So we go on to the next picture. So this is again the same. Now I am giving the color in a different way. Look at this. The floccular nodular lobe and this is the floccular nodular lobe. This is the floccular nodular lobe. And if we say the central part is the nodule, this is the flocculus. They have been marked green. The part of the cerebellum above this is called the anterior lobe, and the yellow part is called the posterior lobe. This is what it looks like. This is a part of the cerebellum. So this part of the cerebellum is actually forming the roof of the fourth ventricle. So this part is the anterior lobe, and this is the posterior lobe connecting this. Now, cerebellar tonsils, I had told you to keep this is the cerebellar tonsil, very close to the midline. And the central part of this vermis, which is of the vermis, is uvula, and the cerebellar hemispheres are tonsils. Why they are called tonsils? Because they protrude like tonsils. On the side of our oral cavity, we have tonsils. They are also protruding like the oral cavity tonsils. And this, what happens when there's raised cerebral intracranial tension, they protrude through the foramen magnum. And this can block the CSF between brain and spinal cord, because I showed you the CSF secretion, because at the roof of the fourth ventricle, CSF trickles down and comes to the subarachnoid space. So this tonsils, when they are pressing against it, it's not going to allow the CSF to come out. It will increase the tension, increase the CSF in the cranial cavity, and cause a condition which is called as hydrocephalus. Sometimes if it herniates outside, it presses against the vitals which are present in the brainstem and cause mortality. It can be fatal. Many conditions happen where the cerebellar tonsils protrude out. That's a syndrome which is called it also happens during congenitally. We see that also when we do fetal autopsies. That's called the Arnold-Chiari syndrome. It can be there present, and they should be always taken care of if it is, because they can lead to very serious conditions. So this is a section of the cerebellum and with the brainstem, and this is only the cerebellum, and you can see that the gray matter is stained blue. This is a section with Toledan blue, and the white matter is white, and this appearance of this branch tree is called the Arbor Vitae appearance. This I've already explained to you. And if you look at the white matter, deep inside the white matter, there's some gray also. That means inside the white matter, there is some gray matter. Those are the deep-seated nuclei of the cerebellum. We'll come to that. You'll be doing a class on histology of the cerebellum where we show you this slide, but just to understand the cytoarchitecture, It's an H&E section where you have the cerebellum is not like the cerebral cortex, which has 6 layers. The cerebellum has 3 layers. The outer layer is called the molecular layer. The middle layer is a single layer of cells which are called the Purkinje cell layer. They are cells, large cells, look like baskets. They are called the Purkinje cell layer and the main cells of the cerebellum. And the inner layer is called the granular layer. So what you see, molecular layer cells are very sparsely arranged with too much of white matter fibers, so giving it a speckled appearance. But when you look at the granular layer, it's a 5-6 layer thick layer with very closely arranged cells which are called the granular cells. And in this you have core of white matter present in the cerebral cortex, and you can see that the cerebellum, these are the folds of the cerebellum which are called folium because they are like leaves. Now if you look at this is a schematic diagram of the cytoarchitecture. The main cell of the cerebellum is the Purkinje cell layer. It's a single cell layer, right? Now, if we look at this Purkinje cell layer, it gives dendrites which are going towards the molecular layer. It gives number of dendrites, a primary, secondary, and then it gives rise to tertiary dendrites, divides, and they give rise to dendritic spines, right? From the base of the Purkinje cell, the axon comes down, goes into the granular cell, goes into the white matter and connects with the deep-seated nuclei of the cerebellum. So these are the Purkinje cell layer and these are the dendrites of the Purkinje cell which run in the molecular layer. So it has multiple dendrites, and look at the shape of this. It's a characteristic feature of the cerebellum. There are number of cells, Purkinje cells, and efferents from the cerebellum only comes out through the Purkinje cells, right? The next cell that we see in the molecular layer are 2 cells. One is called the basket cell and there is another cell, small cell, which is called the stellate cells. These cells send their axons to the Purkinje cells, right? So they are small cells which are just giving the speckled appearance in the molecular layer. An important cell layer which is present out here is the granular cells. In the granular cells, there are number of granular cells. These granular cells, they have number of dendrites. They have number of dendrites, and these dendritic branches, they would be forming number of connections with Golgi cells, and the axons of these would be going out into the molecular layer, and they divide perpendicular, they divide parallel to the folium. They divide parallel to the folia. So you find that the cerebellum has these parallel running fibers, which are all fibers of the granular cells, giving this kind of an appearance. And in the meantime, what you find that these Purkinje cells would be actually synapsing with the granular cell fibers. So what happens is the granular cells which are coming here are giving these bundles, and the Purkinje cell, this is only giving you a schematic diagram of one Purkinje cell, will be all forming synapses with the granular cells. There's another cell out here which is called the Golgi cell. So Golgi cell will be also forming, giving the granular cell some inhibitory impulses by its axon, and the axon of the granular cell goes up into the molecular layer. The dendrites of the Golgi cell goes up into the molecular layer, and these axons out here are all positively charged. The impulses which are coming to the cerebellum are only 2 kinds of fibers, just remember. There are two types of fibers. One is called the climbing fiber. The other is called the mossy fiber. Climbing fiber comes from the inferior olivary nucleus, right? The inferior olivary nucleus fibers are called the climbing fiber. And why they are called climbing? Because they climb into the white matter, the granular cell matter, and then cross the Purkinje cell layer to come to the molecular layer and synapse with the dendrites of the Purkinje cell. So this is what it is. And these Purkinje cell layers, as they are, so you can see them that they would be synapsing, a climbing fiber would be synapsing with these Purkinje cell layers and influencing it, which are coming from the inferior olivary nucleus. The rest of the afferents which are coming to the cerebellum all come by the mossy fibers. So the afferents to the cerebellum are by climbing fibers and mossy fibers. The mossy fibers come and relay in the granular cells, and the granular cells receive from the Golgi cell and send its axons to the molecular layer. And they would be all forming synapses out here. So finally, what happens? The impulses which are all processed in the Purkinje cell body, and it will be coming down and going into the white matter and be going out as efferent pathways from the cerebellum as cerebellar olivary, cerebellar spinal, cerebellar cortical. All these fibers which will be going out are all from the Purkinje cell, going to the deep-seated nuclei and then going out. So one has to understand the two afferent fibers are climbing fiber and mossy fiber. The efferent is coming from the axons of the Purkinje cell, which will be going to the deep-seated nucleus of the cerebellum, and from there they will be going out. None of the other cells give rise to any efferents. They are all giving rise to stimulatory impulses, but the Purkinje cell finally is giving rise to the inhibitory impulses. As I told you from the beginning that the function of the cerebellum is coordinating the posture, the movements, the finer movements of the body, all unconscious proprioception. That is all taking place because it is always under the cortical control. Cerebellum doesn't do it on its own, it's just processing it. So the afferents are the mossy and the climbing fibers, and the nuclei of the cerebellum are dentate, emboliformis, globusus, and vestigial. They are lying in the roof of the fourth ventricle and from lateral to medial. So dentate is the lateral most, emboliformis iske baad, then globusus and vestigial. Afferents are from. And the mossy fibers, when they come out, they form this swollen part, which is called the rosettes, and these rosettes form axon terminals of the Golgi cells. So the Golgi cells will be coming and forming terminals out here to come, and then they would be sending their impulses to the Purkinje cells. So this is called a rosette, and they are all protected by the glial cells, the astrocytes, for conserving the neurotransmitters. So this is what it looks like. This is an axon terminal of a Golgi cell, and the mossy fiber, as it enters the white matter, it gets demyelinated, and the granule cell sends its dendrites. They are forming connections. Axon terminals of the Golgi cell is forming connections, while the dendrites of the Golgi cell goes to the Purkinje cell layer, to the molecular layer. So this kind of structure, one is this swollen part of a fiber is called a rosette. This is called a rosette. It's seen only in the mossy fiber of the cerebellum. Climbing fibers are like a vineyard, climbing, just ekdam like a creeper. They climb up like vines of the grapes in a vineyard. That's why they're called climbing fibers. These are the nuclei. If you take a section out here, This is the cerebellum and brainstem. So in the roof, you find these nuclei. This nucleus is the dentate nucleus. Again, look at this nucleus. It looks like a crumpled bag. And in the hilum of this crumpled bag, the efferents would be coming out. The next nucleus, we have emboliformis and globuses are these small, small round nuclei. And the medial most nuclei is the vestigial nuclei. Now comes part where we divide the cerebellum phylogenetically. Phylogenetically, we are dividing the cerebellum into archicerebellum, paleocerebellum, and neocerebellum. Archicerebellum is the primitive cerebellum, which involves a floccular nodular lobe and the uvula of the posterior side. So flocculus, we have the nodule out here, and so the nodule out here, it's not shown in this picture. So flocculus, nodula, and this is the uvula. ये side में क्या है? Tonsil. So uvula, floccular, and nodule will be forming the archicerebellum. This is a part which in human beings we will find it synonym with the vestibular cerebellum. That means carrying impulses from the sensory impulses from the vestibular ganglion and vestibular apparatus. This is an organ of balancing. That's why it's present in fish and lower amphibians who are swimming, because they need very good balancing mechanisms to know their pathway in water. So their vestibular apparatus is very well developed, and the vestibular cerebellum is developed. That's only part of the archicerebellum. Then came the paleocerebellum with higher amphibians, reptiles, and birds. What happens? This is present in the superior part of the vermis and the rest of the vermis, and the part of the anterior lobe of the anterior lobe also. It is all called as spinocerebellum. So that is spinocerebellum, while neocerebellum is present in higher animals, largest in humans, and the inputs are the pontocerebellar fibers, which are coming from the cerebral cortex. You would have done the descending pathway, the pyramidal pathway, which is the corticocerebellar pathway, corticospinal pathways, and the corticospinal pathway, before coming directly to the spine, they come to the crus cerebelli, come to the pons as the corticopontine fibers, and these corticopontine fibers would be going to the middle cerebral peduncle, coming to the cerebellum to form the corticopontocerebellar, and the cerebellum would be influencing these fibers. If he finds that this movement is not proper, it would be sending its impulses back into the neocerebellum by dentito-rubro-thalamic pathways, which will be again going to the cerebral cortex and then coming down as the extrapyramidal pathways. So they are indirectly influencing the corticospinal pathways. Now, if we divide the cerebellum functionally, so phylogenetically it has been divided into archicerebellum, paleocerebellum, and neocerebellum. Functionally, it has been divided into the vermian part, which is the midline. It influences the long axis of the body. That's the neck, shoulder, thorax, abdomen, hip. That is the vermian part. So any lesion in the vermis would be causing some defect in the midline part. Remember that. There will be some balancing mechanism which will be going haywire in the midline part. Lateral to the dermis is the distal part of the body. That's the hands and the legs and all that. And the lateral most part is planning of the sequential movements, the assessment, conscious assessment of our movements. So the vestibular cerebellar pathways, which are coming from the vestibular ganglion and also from the vestibular nuclei. If you see the vestibular nuclei at the pontocerebellar junction, there are superior, inferior, medial, and lateral 4 vestibular nuclei. It's a big diamond-shaped nuclei. It's a big nucleus, diamond-shaped. These fibers would be forming the vestibular cerebellar fibers, which will be going to the cerebellum, and as they go to the cerebellum, these are the parts which will usually the nucleus for the vestibular cerebellar or archiocerebellum is the vestigial nucleus. And from there it gets influenced and then comes down through the vestibular spinal tract. Sometimes they go directly without even relaying in the nuclei. Then comes the spinocerebellar tracts. You've already done that. The spinocerebellar has two, posterior spinocerebellar and the anterior, or we say the ventral and the dorsal spinocerebellar tracts. They are all this you've already done in the ascending tracts, and you know that the ventral spinocerebellar tract crosses once in the spinal cord and then crosses again once in the go to the midbrain, crossing over, comes to the inferior olivary, where it again crosses and comes to the same side of the cerebellum, while the posterior spinocerebellar is an uncrossed pathway, and that is a anterior spinocerebellar crosses twice. Inferior cerebellar peduncle में the fibers which go are the posterior spinocerebellar pathways. So this is the nucleus which they will be going to, would be the part of the dentate nucleus. They'll be going to the, and the efferents are shown by, afferents are shown by red, sorry, by blue, and efferents are shown by red. So what are they going? They are going to the reticular nucleus. They are going up to the red nucleus. So here when they are coming from the cerebellum to the red nucleus, we will be calling them, or from the dentate, we will be calling them cerebello-rubral or dentato-rubral pathways. And from there, they'll be going to the thalamus. So we say dentato-thalamic pathways, and from there they go to the cerebral cortex, which will be also influenced. So the reticular formation which is there also influences it. So this is the pathways of the, so these pathways would be influencing the corticopontine fibers, which will be forming the spinocerebellar pathways, which will be the cerebellar spinal pathways. So this is finally we say that these are the dentate nucleus and then the thalamus, which I was just saying in the previous picture, come to the pontine and then it will get influencing the corticospinal pathways. This is this picture you must have already seen when you did the anterior and the posterior spinocerebellar pathways. So I'm not going to go into the detail of this, but you have to learn these pathways very well. So what is it? What is the what is it doing? What is the cerebellum doing? It is processing. So from the muscle spindles, it takes the afferents, goes to the cerebellum, then it gets controlled by the cerebral cortex. Commands, no, it doesn't lie. This is not the movement to be done. This is the movement to be done, or whatever instruction it gives comes to the cerebellum, and then the feedback mechanism will be such that again it will go up to the cerebral cortex to give the appropriate function. So this is fourth ventricle. We'll be doing the fourth ventricle when we in a demo and show you the parts. But you can see this is the floor of the fourth ventricle, which is formed by the upper part of the medulla and the pons, and these are the peduncles that you see, and the roof is formed by the cerebellum. Above we call the cerebellum the thin matter, white matter that is here, we say it's called the superior medullary vellum. Inferiorly, similarly, there is a white matter which is called the inferior medullary vellum. So these are thin layer of matter which are forming the roof, and you can see that the central line is called the sulcus limitans out here. This is the median line, and the sulcus limitans is actually the one which is separating the motor from the sensory area, right? And can you see these fibers, strands of fibers which are there, cutting this into two triangular area, an upper and a lower? These fibers are called the stria medullaris. So this is a part of the brainstem showing you clear cut section. You can see the pineal gland with the superior inferior colliculus. This is part of the thalamus and this is the part of cerebral cortex. So we'll be doing that. So the medial zone, as we said, that is for the midline, is for the spinocerebellar tracts. The flocculonodular lobe with the lingula, it's for the vestibular cerebellar. Intermediate is the pontocerebellar, and this part is coordinating. It does all the processing. This is the way the cerebellum acts. So when we look at the cerebellum, it has first a morphological division by the primary fissure into an anterior lobe, posterior lobe, and a posterolateral fissure, separating it into a flocculonodular lobe. It has a phylogenetic classification as Archicerebellum, Paleocerebellum, and a Neocerebellum according to connections. And then the body is represented in the median part, intermediate part, and lateral part is the process. That means you can understand that cerebellum has an important role in carrying out all our motor functions. So any midline swelling would call the like a medulloblastoma, or chronic alcoholism also causes degeneration of the bones, where the person would be having an ataxic gait. What is an ataxic gait? Well, if possible, we'll try to see. We'll ask neurology to show you some videos on this, where the person has a waddling, staggering gait, can't walk properly, and to compensate his walking imbalance, the swinging or swaying movement, he has a wider base. नहीं तो फिर strides ठीक नहीं हो तो person can fall. Nystagmus is the jerky movements of the eyeball when one wants to focus on a certain object on one side. There is flickering of the eye movement, jerky eye movements. That's usually when there is an midline involvement. Lateral involvement leads to ataxia, that is again a clumsy movement, dysmetria. And this leads to pass pointing, is a function by which you ask the patient to touch his nose. He will not be able to touch the nose with the finger. With the finger, if you ask the patient to close his eyes and touch his nose, it will be overshooting, going somewhere else. It will not touch the nose because there is no coordination between the muscles of the hand and the brain. Thereby, it will be going here and there, which is called pass pointing. Clumsy, rapid movements. Movements, if you ask a person to do supination, pronation, he will not be able to do it. That is called dysdiadochokinesis. Asynergia, no tone in it. There's no systematic movements. Hypotonia, very, the muscle tones are not there. They're flaccid. And they'll be static and intentional tremors. There'll be tremors. Tremors means very, we will say movements, fast movements which will be present without even doing any action. Dysarthria, the difficulty to speak. So all this would be there. And this is just a problem. Instead of starting the class with a problem, I've given you this problem later and we'll be sending you SQLs and some questions, because cerebellum is an important topic and slightly difficult because not like the straight tracks. So this is a boy, 4.5 years, was taken to the neurologist. The mother was very bothered that the patient is having a lot of vomiting on waking up in the morning and unsteady gait while walking, while standing up, and usually falling backwards. Physical examination, the child tended to stand with the legs well apart. That's a broad-based gait. Head was larger than normal for his age, and the suture lines of the skull could easily be palpated. That means what happens? The tension of the intracranial tension has increased. Retinal examination showed papilledema of both eyes. This would be done by an ophthalmoscope to see the fundus of the eye, and there is edema in the fundus. Muscles of upper lower limb showed some degree of hypotonia. Nystagmus was not present. That means the child had no nystagmus and no tendency to fall to one side when asked to walk. So using this knowledge of neuroanatomy, explain this lesions. Is this lesion in the cerebellum like to be in the midline or to one side? So I've given you the answer. This is a medulloblastoma. It's a midline tumor which is affecting the midline of the body. That's why when he is walking, there is an unsteady gait, but he's not swaying. He's swaying, but he's not going on to one side. If it would have been a lateral tumor, it would be going on to one side. But as the tumor increases, all the cerebellar involvement will take place, and that is the reason why the head is larger and the skull sutures are separating out. That gives an idea that this is some kind of a tumor is present in the brain, in the brainstem, that's near the medulla, because that is where it is pressing against the vermis and then the cerebellar hemisphere, which is called a medulloblastoma. So thank you, and I'll take the next class I'll be taking will be on thalamus. Somebody is asked about term exam. We'll let you know about the term exam once we have a meeting. It will be decided. But first of all, let's finish the course and let's give you some kind of revision before we do this. Before we have any term exam, we will discuss with your other departments and then decide. Because it's too early, let's see what happens. उसके बाद हम decide करेंगे. And anyone wants to ask something? Gauri, are you there? I'll just try to unmute you.
Good afternoon, ma'am.
Good afternoon. I've just unmuted you. So as soon as I get the orders and all that from the dean, I'll send it to you. You can send it to the class. And
Yes,
any questions regarding neuroanatomy, let me know.
Yes, ma'am.
आज का क्लास समझ आया?
Yes, ma'am.
या कुछ चीजें, because ये थोड़ी, this is a slightly different classes anyway. And I get a feedback from, this is for everyone, not only for the CR, because she's representing all of you, she gets the brunt of it also, that you people are not submitting your Google forms. So that's not done. Please submit. Last time there were only 200 forms came, and yesterday again there was a form sent. The Brainstem, has it gone? Yes or no?
Yes, ma'am. Yes, ma'am.
When did you get it? Today?
No, ma'am. Yesterday around 8:30, ma'am
Okay,
sent it
so
and said it's open till 11.
Okay, so I get the feedback about that also, and then we'll send we'll send Google Docs on this also. But the thing is that you should be also being responsive in doing
Yes,
whatever we are doing, right? So that's it. I'll get back to you once I have some idea feedback.
yes, ma'am.
Okay, and
Yes,
you
ma'am.
also let me know how, but I know that many of you are staying in the hostel also. So
Ma'am, there are a few students.
there are a few students, right? So, okay. So, bye.
Yes, thank you, ma'am.
Thank you.